AI minds, quantum leaps. Cut the noise, dive deep. Truth unlocked. Dr. Mike on the mic. Tech Unfiltered is on the clock. Now it's time for the show.
Here's a sentence I did not expect to say this soon. A computer wrote a virus. Not a computer virus, but a biological one with DNA and a protein shell. Human researchers built it in a laboratory, and it worked. That opening needs an immediate qualification. These were bacteriophages, usually called phages. They infect bacteria, not people. The experiment used harmless laboratory strains of E. coli under containment. Nobody created a human virus, and this study does not show that a chatbot can casually design one. With that clear, the result is still remarkable. On August 6, 2026, researchers led by Stanford and the Arc Institute published a paper in Science describing the first generative design of complete viable phage genomes. AI has been used in biology for years to predict protein structures, design an enzyme, or change one component. This time, the models generated the entire genetic instruction set. The researchers used systems called EVO1 and EVO2. They are genome-language models. A text model learns patterns and sequences of words. A genome model learns patterns and sequences of DNA. In this case, the model was asked to generate a full viral genome rather than one isolated part. The target was a well-studied phage called phage-174. Its genome is only about 5.4 kilobases long, which is tiny by genome standards. But tiny does not mean simple. Its genes overlap, regulatory elements interact, and one genetic change can disrupt several functions at once. It is a compact, difficult test of whether a model can coordinate an entire biological design. The team fine-tuned the models on nearly 14,500 related phage genomes. The systems produced thousands of candidates. Researchers selected roughly 300, had their DNA made, introduced those sequences into bacterial cells, and tested what happened. 16 designs produced functioning viral particles. They could replicate and kill their target bacteria. 16 out of roughly 300 is about a 5% success rate. That is inefficient, but the important result is that the number was not zero. Software generated a complete genome, people assembled it, and the resulting biological system performed its intended function. It is also worth being precise about what the AI did and what people did. The model did not choose a medical problem, decide to create a virus, order genetic material, or operate laboratory equipment. Researchers selected the target, prepared the training data, defined the design constraints, chose which outputs to build, and performed every physical experiment. The model's contribution was generating candidate sequences that coordinated all the necessary genetic parts at once. That is a major technical achievement without turning the software into an autonomous biologist. You may also hear this described as AI creating life. That phrase is more dramatic than the result supports. Scientists still debate whether viruses count as living organisms because they cannot reproduce without taking over a host cell. The narrower and more defensible claim is powerful enough. AI generated complete genetic blueprints that human researchers converted into viable, replicating viruses of bacteria. This distinction matters because it tells us where responsibility still sits. The generated sequence does not walk out of a computer by itself. It has to pass through people, funding decisions, research review, a DNA manufacturer or synthesis facility, and a laboratory. Those steps are not reasons to ignore the capability. They are places where society can inspect, slow, or stop unsafe work, while allowing legitimate medical research to continue. Some generated phages perform better than the natural reference phage in laboratory growth competitions or killed bacteria faster. A cocktail of the generated phages also overcame resistance in multiple E. coli strains that resisted the reference phage. Under a cryo-electron microscope, one generated phage appeared to use a DNA packaging protein from a distant viral relative, a combination that previous human design attempts had not successfully produced. Why make new viruses on purpose? The medical answer is phage-therapy. Antibiotics attack bacteria chemically. Phages attack bacteria biologically, and they can be highly specific about which bacteria they infect. That specificity may help when an infection no longer responds to antibiotics. It also creates a challenge. Bacteria can evolve resistance to phages, so clinicians may need a new phage or a new combination quickly. Today, finding useful phages often involves collecting environmental samples, testing many candidates, and hoping one matches the bacterial strain. If genome models can generate diverse candidates on demand, part of that search might eventually become a design process. That is a projection, not a clinical result. Nobody was treated in this study. All testing occurred in laboratory dishes. The work suggests a possible path toward more adaptable phage therapies, but it does not prove safety or effectiveness in patients. The researchers also built safeguards into the work. They used non-pathogenic bacterial hosts and dedicated containment procedures. More importantly, the training pipeline deliberately excluded sequences from viruses that infect humans, other animals, or plants. ARC Institute says its red team testing found that attempts to generate pathogenic viral proteins produced effectively random output. That supports the claim that this system, in this configuration, was not capable of designing a human pathogen. Those precautions are real and important. They are not a permanent guarantee for every future model. More complex viruses have larger genomes, more complicated biology, and many barriers between a sequence on a screen and a viable organism. Synthetic genome engineer Tom Ellis told The Guardian that this was the smallest and easiest kind of viral genome to make. He also noted that modifying a known pathogen remains a more accessible threat than designing a new one from scratch. The concern, then, is not that this experiment secretly produced a pandemic virus. It did not. The concern is the direction of the capability. A reusable model has moved from proposing biological pieces to writing a complete genome that became functional after expert human assembly. In a companion article in Science, health security experts at Johns Hopkins praised the team's precautions, but warned that governance has not caught up with the ability to compose viral genomes using generative AI. That gap matters because many existing safeguards look for known dangerous sequences. A DNA order can be compared with the lists of recognized pathogens or toxins. A generative model may produce a sequence unlike anything in nature while still preserving a biological function. A simple matching system may not recognize that risk. The strongest response is layered protection. A biosecurity expert at King's College London argues for safeguards during model development, responsible review of proposed research, screening when DNA is manufactured, laboratory biosafety and biosecurity, and surveillance for unusual biological activity. No single checkpoint should carry the whole burden. There is a defensive opportunity here too. Better genome models could help scientists design targeted phage cocktails, anticipate bacterial resistance, build countermeasures, and improve screening. The same speed that creates dual-use concern could strengthen our defenses if safety tools advance alongside design tools. For ordinary people, this is a preview of what changes when AI moves from information into fabrication. A text model produces an answer. A genome model can produce instructions that, after substantial human expertise and physical synthesis, become a biological object. The human work is essential, but the digital output can now guide something that exists and replicates in the physical world. The balanced conclusion is neither panic nor dismissal. This experiment may contribute to treatments for infections that no longer respond to antibiotics. It also shows that whole genome generative design is no longer hypothetical. We should protect the medical opportunity while building safeguards at the model, research review, DNA synthesis, laboratory, and surveillance stages, while the capability is still limited and difficult to use. Until next time.